Bismillah, Assalamualaikum warahmatullah. You're listening to Immunology lesson on serology and other immunologic techniques. The learning outcome for this one would be a basic familiarity with the common techniques used in immunology. At your level, as a piece of pragmatic advice, I wouldn't worry too much about memorizing every little detail of every individual technique that currently exists, because when you work later or when you do your PhD, you won't use most of the techniques out there. So your aim here should be to get a bird's eye view of the common techniques. So that will become sort of a launching point later when you need to use specific techniques for your PhD experiments or your lab task at your workplace. And then when you get there, when you really need it, then you can go crazy into details of these techniques. So serology simply means the study of serum, which is the portion of the blood that you can get after you spin it down through centrifugation. So it's the portion that excludes erythrocytes, for example, the red blood cells, and the clotting factors. Inside the serum, you've got a lot of antibodies that you can utilize for diagnosis. So some of the techniques include radioimmunoassay and ELISA. In radioimmunoassay, we tag the antigen, meaning we stick them with radioisotopes or radioactive atoms. So we call this radio labeling. And then we use a machine to detect the radioactive decay of those atoms. And that becomes an indicator of the level of antigens or antibodies that we are interested in. Now, although radioimmunoassay is highly sensitive, it's also quite pricey, so most of the time we don't use it for our immunology assays. Instead, you're more likely to work with a technique called ELISA. So there are many variations of this technique, but the principle is similar enough that if you understand one, you kind of understand them all. So we'll just look at the common one, which is the deliciously named sandwich ELISA. The reason we call it that is because you know how in a sandwich, we got our salad in between two slices of bread? So in sandwich ELISA, we have our antigen in between two antibodies. The way you do it is first you take your microtiter plate and you pipette in your first antibody solution. We call it our primary antibody. Then we incubate it for some time so that our primary antibodies stick to the plate surface. So that's our first slice of bread. And after that, we put our antigen in. We incubate it again. So if the antigen matches our antibody, it's going to attach to the antibody. And then you wash the wells to remove the antigens that don't get attached. So what you do is you put a couple of layers of paper towels on your lab bench, and then you turn the plate upside down and then you bang it onto your lab bench, like bang, 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 because you really want to get rid of the unbound proteins. So they won't be there when we run the plate in our ELISA machine later. So you've got your primary antibody, then you attach your target antigen, and now you put your second slice of toast, of bread, to complete your sandwich, meaning you pipette in your secondary antibody. And then you incubate it to let the antibody bind to your antigen. And then you wash it again. You pour in your buffer solution, you turn your plate upside down, and then you bang it on your lab bench. Hard. I mean, don't be shy about this. And so if you happen to just join an immunology lab and you see a senior PhD student banging her plate like crazy on the bench, she either just had a frustrating meeting with her supervisor or she's just doing ELISA. The reason we do this is that in the end, we will run our plate in an ELISA machine. And that machine doesn't detect the antigens directly. Instead, it detects your secondary antibody because that's where your fluorophore is. The fluorophore is a chemical fluorescent. So think of it like the shiny part of your antibody-antigen complex. So it's the part that your ELISA machine can see. So essentially, the machine doesn't see the antigen. It sees the secondary antibody. So if you don't wash your wells properly, you will have too many secondary antibodies, more than the amount of antigen there is. So if that happens, the machine will miscount the amount of antigen that you have in your sample. Otherwise, if you do your ELISA correctly, you will have a good curve, a good pattern of antigen concentration detection. Another technique is called immunoprecipitation. So here we use the antibody to precipitate or to concentrate our target antigen. So imagine you have a sample that consists of many types of antigens and you want to detect one type of them. What you do is you add an antibody that has been attached to a bead. So some of these beads are magnetic and some are just organic beads like agarose. So it depends on the immunoprecipitation protocol that your lab is using. So these beads are attached to the Fc region of your antibody. So if you can't remember what Fc and Fab regions are, listen to my previous episode on antigen recognition molecules. So you add in this antibody and the Fab region of it will attach to your target antigen. And then you spin the tube down. What happens is your complex of beads plus antibody plus antigen, this complex will have a density, the heaviness, that when you centrifuge the tube down, the complex is going to end up at the bottom. And the antigens that you're not targeting for, they will end up floating around the top. So the next step is obvious then. You simply wash out the antigens that are not your target antigen. And finally, you elute your antigen, meaning you extract your antigen away from the antibody. Another technique is immuno-affinity chromatography. A quite similar principle, but this time your beads and antibodies are structural parts of the chromatography column. So you get yourself a chromatography column. So it's a device shaped like a column, that long vertical structure. Like if you get to visit Trafalgar Square in Westminster, you can see this tall column surrounded by lion statues around it. So this chromatography column, it has antibodies and beads embedded inside it. What you do then is you slowly flow your sample through the column. So your target antigen then will stick to the antibody. And then you wash away the unbound proteins that don't match your antibody. And finally, you elute your target antigen. You can, for example, change the pH condition or wash the column with some chaotropic agents, meaning chemical agents that can disconnect the binding between your target antigens and antibody. Another technique that's commonly used in immunology experiments is called Western blotting. With this, you try to determine the relative quantity of the protein present in your sample. So if you've got your experimental group, you want to know whether it has more protein than your control group. It has 4 steps: sample prep, gel electrophoresis, membrane transfer, and immunodetection. The first step is sample preparation. You take your samples, you put them in a buffer solution containing SDS and mercapto, and then heat it for 5 minutes at 95 degrees. Actually, depending on the sample, sometimes you have to do other stuff first, like sonication, which means you use the sound wave to crush your cells first. So whatever the steps are, they are meant to break down your cell structure and expose your proteins. The next step is, after you have your protein sample prepared, you run it through gel electrophoresis. When you work at a job later, or when you do your PhD, your lab usually has an established protocol for this. So how much sample should you put in, how much voltage, usually 100 to 200 volts, and how long you should run it. But in general, what electrophoresis does is it separates your proteins into bands, like dark lines. So each line represents a cluster of proteins of certain size. And then after you got your proteins separated on the gel, the next step is migrating those proteins onto a membrane. So a membrane looks like a sheet of paper. What you do is you sandwich them. You lay the membrane and the gel in the middle, and you put filter papers and pads around them too. You prepare your sandwich in a buffer, by the way. You don't want it to get dry. And then you put the whole thing in the machine and you run it. So what happens here is at the macromolecular level, your proteins will be pulled from the gel to the membrane. After the machine is done, you take the membrane out. You can check out the gel because the proteins are gone there. The proteins are now on the membrane. So here we do what we did with the ELISA, or something similar. We pipette in our primary antibody and we put in some blocking buffer like milk or albumin. What we're doing is we try to stick our primary antibody on our target protein. And the milk, the blocking buffer, we use it to block the other proteins that we don't want to detect. So we incubate it for an hour or overnight, depending on the protocol or how finicky your target protein is. And then you put away the excess solutions, and then you pipette in your secondary antibody. It will bind to your primary antibody. So the secondary antibody then will luminesce, meaning it will show up on the machine. So basically, the logic is the more intense your luminescence is, the more proteins are present. So the darker the band, the more target protein you have. So you can then use a program to analytically compare the intensity of the bands between your samples. The next technique we're going to look at is flow cytometry. So remember I talked about biomarkers in one of the previous episodes, like CDs, clusters of differentiation. So those are some of the things we can detect with flow cyto. We can detect molecules expressed on a cell surface or even inside the cells. So we do it by staining the cells with compounds called fluorescent probes. So they fluoresce, they absorb light and release it at a longer wavelength, which our machine can detect. So the machine can detect the probes and count how many you have in each sample. And not just that, the machine can also sort out the probes of different fluorescent wavelengths. So to make it simple to understand, think of it like this. The probes are like balls with different colors. So different balls will bind to different biomarkers. So the machine can sort out the balls. It can tell you how many balls you have for each color. So that machine is called a fax machine. So the fax machine is expensive. When I was studying, the uni even assigned a technician with a PhD to take care of it. So it's expensive because it's powerfully sensitive. I'll leave links below if you really are interested in how exactly fax machine works. But the basic idea is, firstly, you treat your cell samples with probes, multiple probes which can detect multiple biomarkers. Once your sample is prepared, you put your tubes in the machine. The machine will drop your sample super fast as a thin flow, very thin, like one cell at a time, theoretically. The machine then will fire a beam of light towards the cells. So depending on the properties of the cells, the light will scatter in different manners. The machine then has different detectors to capture those scattering patterns. And then the machine will sort out the signals of different probes and count them. And of course, the machine will feed all those signals into data points on a computer. So for example, if the forward scatter pattern has higher magnitude, the machine interprets it as your cells are bigger, things like that. So in the end, you're going to get quite a detailed profile of your cell sample population. All right, let's end this episode with an easy one, hemagglutination. So this is the technique you would most likely be familiar with, even if you don't take immunology. We use this technique to identify the ABO blood group. So this is extremely useful during a blood transfusion. The principle behind it is that people who express A or B blood group antigens are tolerant to those antigens, and individuals who don't, they produce antibodies that attack those antigens. So before a patient receives a blood transfusion, we've got to look at their ABO type. If they have type O, for instance, the patient will have natural antibodies against A and against B. Therefore, if you give her blood from type A, type B, or type AB donors, then the patient's immunity will attack that donated blood. So the blood will agglutinate or clump together. So type O patients can only receive type O blood transfusion. Now, ABO grouping is not the only thing that matters immunologically. We also look at patients' rhesus type. That's RHESUS, rhesus. This matters, for instance, if you're a woman who has rhesus negative and your husband has rhesus positive, and then you get pregnant, and your first baby gets the rhesus antigen from your husband. Your baby has rhesus positive blood. What may happen is that your immune system might slowly develop an antibody against that rhesus positive blood. So the first baby will be okay typically because the immune sensitization takes time. But by the time you get pregnant with the second baby, your immune system may have fully raised an army of antibody against rhesus positive blood. So say your second baby again happens to be rhesus positive, right? They want to be like their dad. So if the second pregnancy involves a rhesus positive baby, then there is a good chance that your immunity will attack the blood of the baby. What do we do then? What we do is we give you what's called a ROGAM injection. So it's given intramuscularly, meaning you'll get injected on your bum, your glute muscle. So the ROGAM injection will be enough to protect your baby from the hemolysis, from being attacked by your body. So it's quite rare. Rhesus negative is rare. Actually, the rarest one would be people who have rhesus null. So I learned that from Grey's Anatomy. I'll try to find the link to that episode and put it below. You should watch it. They call it golden blood. There are about like only a few dozen people who have been recorded to have rhesus null. I mean, out of 7 billion people on the entire planet, right? So while rhesus negative people don't have what we call a rhesus D antigen, rhesus null people don't have any rhesus antigen. So it's just so rare for that to happen. I'll leave links about this stuff later for you to explore. Okay, that's it for this topic. Talk to you next time. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.